So now we're going to get into saponifiable complex lipids. Complex lipids. Now, let's go ahead and go back a couple slides. And let me bring this point home. In this case right here, this is an example of a overall simple lipid, such as a triglyceride. Why? Because it has 3 fatty acids. So we can say that this is simple. Compare this picture now, where although we do have 2 fatty acids, we have a brand new 3rd type of non-fatty acid. This non-fatty acid can be some other type of molecule. In this case, in the categorization of phosphoglycerides, 2 of them are fatty acids, and then the 3rd tail is actually going to be a phosphate derivative. So I'll write this here, a phosphate derivative. So we say that this is going to be a, and I'll circle this up in the upper left corner, complex lipid. Because although 2 of them are fatty acids, the 3rd one is going to be a non-fatty acid, and depending on what category you're in, it will vary. So in this case, for the complex lipids, it's going to be a, in this example, it's going to be a phosphate group, so we call it a phosphoglyceride. The phosphoglycerides, again, are made up of the glycerol, 2 fatty acids, and then a 3rd non-fatty acid we call a phosphate derivative. Very, very simple. We have a phosphate group, PO4, that's connected, and then we have one of 3 type of molecules. We can either have a choline attached, a serine attached, or a ethynylamine. These are the 3 type of molecules that could be attached, and it dictates what type of phosphoglyceride we're dealing with. So let me go ahead and show you this. I've handpicked these just to try to make memorization a little bit easier. Look over here. This is a ethynylamine. It contains a alcohol on one end, and it contains a ammonia on the other end. And notice there's 2 carbons in between. Compare that to a choline. Notice you have an alcohol on one side, and on the other side, instead of a traditional amine for primary, you'll actually have 3 methyl groups that are attached. So we actually call this, the fancy word for this is a tertiary amine, actually a quaternary amine, excuse me. So that's the main difference right here between a ethynylamine and a choline. So let me go ahead and re-summarize that. In the ethynylamine, it looks like this, where you'd say where it ends in 3. It ends in 3 hydrogens, whereas a choline, it actually has 3 methyl groups. So that's the big difference between choline and ethynylamine. If we take a look at serine, here we still see the same 2 inner carbons and the alcohol. Over here we have a NH3. However, we now have a carboxylic acid that's attached. So you're responsible for memorizing these 3 initial structures: serine, choline, and ethynylamine. This is important because I'll ask you to actually make a phosphoglyceride. If it contains a serine, then we call this a phosphatidylserine. If it contains a choline, then we call this a phosphatidylcholine. And finally, if it contains a ethynylamine, what do you think we call it? Yes, a phosphatidylethynylamine. You have to memorize their general properties. These are essentially cell membranes. So phosphoglycerides, they make up the majority of cell membranes for all cells. In this case, phosphatidylcholines are the main component of cell membranes. Obviously, the phosphate group makes it very polar heads on the triglycerides, so they're going to be facing toward the water. And the nonpolar tails are going to be on the inside of the membrane itself. And I'll go into detail on that in a bit. They also create a great emulsifier. So we've already talked about emulsifiers, we just never gave it a name. Soap is a great emulsifier because it allows you to link together dirt on one side and water on the other. So in this case, although it wouldn't be dirt, it would be other nonpolar molecules, and we'll investigate those at a later time. If you have phosphatidylcholines, we have a subcategory for these. We call these lecithins. If you have phosphatidylserine or phosphatidylethynylamine, we tend to find these in neural tissue. So think of these as part of the neurocell tissue or brain tissue. So the fancy word for these are cephalins. Cephalins. You're responsible for that terminology. Let's go ahead and show you a chemical reaction. In this case, we have we have two fatty acids. We have a glycerol molecule, only because this is a complex triglyceride. We now are going to add in the phosphate group here, and then we're going to add in and it's going to be one of the three. In this case, we add in choline. Let's go ahead and take a look at the linkages. In this case, the phosphate and the alcohol portion are going to undergo a esterification. Because phosphates have multiple groups that can esterify, notice the other side will also form a esterification. So you have two esterification reactions which are going to occur. Because we added in a choline along with a phosphate, we cleverly call this a phosphatidylcholine. And yes, you will be asked to sketch these. Here is the other derivatives, phosphatidylserine and phosphatidylethynylamine. You will need to memorize and sketch these phospholipids. So usually I give you the option of sketching with any type of fatty acids you desire. And my advice is, if I give you that option, use the easiest ones. In this example, I went ahead and I have two. Let's see, what are these? 1, 2, 3, 4, 5, 6, 7, 8. 2, 4, 6, 8, 10, 12, 14, 16, 18. So these are steric acids. So notice I've drawn the easiest ones, the saturated ones. In this case, 2 steric acids. So this part you're going to spend a lot of time on. If I ask you to sketch a phosphatidylcholine, then you're going to go ahead and sketch something very similar, only you're going to remove these methyl groups and replace them with hydrogens. If I ask you to go ahead and draw a phosphatidylserine, then you're going to go ahead and remove the hydrogens, and you're going to go ahead and add in a carboxylic acid right here. And then they summarize it as a COOH. So memorize and sketch these phospholipids. I love asking these as general questions. Here are examples right here of how I could ask you to sketch these and practice sketching these, as well as naming them. Let's go ahead and move on. We tend to find phospholipids, again, in cell membranes. They are made up of a double layer. So this is a double layer. Notice the tails are going to be facing on the inside. So the hydrophobic portion, or aka nonpolar section of the tails, will be on the inside. And then the hydrophilic heads, the polar section, is going to be facing outward. So on a cell, on the inside of the cell, it's very aqueous. On the outside of the cell, it's very aqueous. So this is why we have a double layer, so that on both sides, it's going to be facing water. Here are general terms you need to know. Prokaryotic cells contain no nucleus. They do have a cell membrane, but no internal membrane structures. Eukaryotic cells contain membranes, both cell membranes, and they contain organelles. And each one of these has a membrane composed of the double phospholipids, as you see down here. So make sure you know these general terms. If you've taken biology before, this is fairly straightforward. If not, make sure you memorize. Memorize, memorize, memorize. We call this the fluid mosaic model. And basically what happens is it's a sea of moving phospholipids. So it's not a true solid. And basically we have water molecules on each side, and they help maintain this structure. But what I often like to do is I often like to think of this as if I were to sit there and I could sit there and have a aqueous solution, and then I might have a layer of fat, and this would be a non-aqueous in between. And then I might have an aqueous solution on the other side. So in the middle we would have a suspended non-aqueous fat layer, so to speak. So think of this cell membrane as floating on the inside of water molecules, and it's maintained, this sandwich, so to speak, is maintained by oppressing water molecules on each side. Do you need to know these explicit details? No, you don't. However, you do need to know the overall fluid mosaic model. Memorize this definition. We have a ton of other molecules, biomolecules, which are embedded into the phospholipid bilayer. So they're embedded on the inside. We have proteins. The proteins themselves can be types of enzymes where reactions can occur. You can have antennas, carbohydrate antennas on the protein, and they're just like real radio antennas. They're used for cell-to-cell communication. So you have various types of lipids, carbohydrates, and proteins that are embedded in the bilayer. Memorize this general definition, be able to explain a quick little explanation of it, and let's move on. These are called sphingolipids. Oops, let me go back. These are called sphingolipids. They're another type of complex lipids. Now, if we've been messing around with the glycerol head or glycerol cap, which binds to the fatty acids and the phosphate derivative. In sphingolipids, basically what we do is we replace the glycerol cap with a sphingosine cap. And this very ugly looking molecule right here is called a sphingosine. So let me go ahead and highlight. This ugly molecule right here is called a sphingosine. What I've done is I went ahead and I highlighted it right here in this example right here. This is a sphingosine. The sphingosine, we now have a brand new category because of this sphingosine. We call these sphingolipids. These are a type of complex fatty acids, replacing the glycerol with sphingosine, hence sphingolipids. I've said that twice now. And this is a type of sphingolipid called a glycolipid in this example down below. So, this type of sphingolipid called a glycolipid gets its name because here we have a carbohydrate, hence the glyco portion. So what do you need to know? You need to be able to recognize. You don't have to memorize the structures and sketch them, but if I give you this structure right here, you're going to have to recognize. So again, recognize. Can you say that again? Recognize. You need to recognize these structures. Immediately be able to classify it. We have two types of main classifications of sphingolipids. We have glycolipids and we have sphingomyelins. A sphingomyelin, I'm going to use my highlighter, contains a phosphate and choline group. So the minute you see this part right here, recognizing it, you can give the name sphingomyelin. It's a nice insulating sheath that's applied on nerve cells to make sure you maintain your voltage on the nerve cells. So think of it as a rubber covering on a wire, so to speak. The other type are glycolipids. You just saw one earlier. And the minute you see a sugar molecule attached to a sphingosine, then you will automatically, and here is my sphingosine right here. The minute you see this present, you're going to name it glycolipid. The alternate name is cerebrosides, and these are other types of biomolecules found in brain tissue. You don't need to read too much into that. Just be able to recognize and generally give a general property of them. I've given the general properties. Sphingomyelins, they tend to be myelin sheaths found in nerve cells. Glycolipids, they just tend to be found in brain tissue. That's as much as you need to know. Since we have so much to learn, let's move on. These are non-saponifiable lipids. Non-saponifiable lipids tend to contain this ring system that you see here. This is called a steroid ring system. So now we're getting into steroid molecules. Notice this is non-saponifiable. Why? Well, do you see a ester present? The answer is no. Consequently, this is a non-saponifiable lipid. Steroids contain the steroid ring system, so now you know from a biochemical point of view where we get the name steroid. And it's a broad class of compounds. It contains a 3 6-membered ring. Here are the 3 6-membered rings in this form. And a 1 5-membered ring, all fused together. So here is a classic steroid. And you probably never knew this, but technically cholesterol is a steroid. So in this case right here, this makes up the parent portion of the molecule. Notice we do have functional groups that are coming off. So this is the most abundant steroid in the human body, essential component of cell membranes. Cholesterol helps make cell membranes a little bit more flexible and fluidic. So we tend to have those embedded in cell membranes. So you're responsible for knowing general properties. So memorize a couple of these properties. I could ask you, name a purpose of cholesterol, name general properties of a cholesterol. When you have too much cholesterol present, we don't know what to do with it, so we tend to store it. Of all the wrong places, we tend to store it in the arteries. Consequently, you have too much buildup, and it causes atherosclerosis. Am I going to ask you to draw a non-saponifiable lipid? No. Bottom line is you're going to need to recognize. So if I give you a structure like this, recognize that this is cholesterol. And of course, name 1 or 2 properties. Hey, look, this is a non-saponifiable lipid. Why? Do you see any esters on here? No. In this case right here, this is a steroid because we have a steroid ring system. Here's an example of a amphipatholic. Amphipatholic is a fancy way of saying having both polar and nonpolar. Consequently, it can bind to both water and nonpolar lipids. Now, when you combine to both water and nonpolar lipids, we do have a fancy word for it. It's just like soap. And because it can bond to 2 different things, we call this a emulsifier. An emulsifier. So it's actually used as a middleman, helping to digest fats, because it allows it to clump together around the insoluble fat molecules. Consequently, we tend to pump this into our stomach when we're dissolving fatty foods. So, bile is used as an emulsifier by the gallbladder, which is emptied into the intestine to aid fat digestion. So, bile contains cholic acid, and cholic acid is a emulsifier. Notice it has the nonpolar portion, and this is where the fatty acid tails, fatty acid tails like to stick. Over here we have the polar portion. The polar portion is the portion which likes to stick to the solvent water. I think I've beat this to death. Bottom line is memorize this slide, and above all, recognize glycolic acid. Steroid hormones. In this case right here, hormones are the chemical messengers. I do want to point out, if we're dealing with a steroid hormone in a biochemistry setting, you know that the hormone is going to contain, and you go back a couple slides, this ring system. In general, think of hormones as these molecular messengers. And basically, they tell the cell what to do. So in this case, here you have a cell, and it's going to go ahead and communicate to another cell. So it's going to go ahead and create a hormone receptor. I'm sorry, it's going to go ahead and create a steroid hormone. The hormone is going to go ahead and come on in to the cell. It's then going to go ahead and check in at the desk, so to speak. And the desk is going to be a steroid receptor. It's kind of complicated, but it relays its information to the steroid receptor, which directly goes to the brain or nucleus to make the necessary changes. So they're the chemical messengers. You don't have to go into any more detail than what you see on the slide. Multiple steroid hormones bind to DNA in multiple cells, affecting cell performance, reproduction, and replication. There are two categories of steroid hormones, adrenocorticoid and sex hormones. So, the adrenocorticoid, you again have to memorize these names. We have mineralocorticoid and glucocorticoid. Here are two examples I could give you. Recognize the structures. I'm not going to ask you to sketch them, but recognize the structures. This is aldosterone. Mineralocorticoid, think the name mineral. It affects the absorption of minerals, aka salts, such as sodium chloride. So, that's an example of a mineralocorticoid hormone. So know the function of it. Regulates the concentration of ions, aka salts, and recognize the molecule that I could use to test you with. Here's a glucocorticoid. Whoa, gluco enhances the carbohydrates, gluc. So make that connection. And this is what cortisol looks like. So it's a steroid hormone, aka adrenocorticoid hormone, when you classify it. This is called cortisol. So memorize the name, recognize the structure with the name, as well as its overall properties. So memorize the names, the properties, the classes, and when it comes to the molecules, be sure to recognize the molecules so that way you can relate the information as you see here. Sex hormones. These are my two favorite ones. There's a ton more, but these are the ones to make your life a little bit easier. This is testosterone. Notice it has a ketone in it, so that's the trick on this. These were found in the testes, so that's where they came up with the name testosterone. Female sex hormones. Here's a nice example of one. We call this estradiol. They got the name diol because it has two alcohol groups on there, and it makes up estrogen. So biochemistry, they just come up with any reason whatsoever to come up with the name. There really is no systematic function. That's why you have to memorize these names. So compare these two molecules. They're both steroids due to the ring structure. But again, notice the key differences between the two. So if you see a ketone and alcohol on this structure, testosterone, but if you see two alcohols, then it's estradiol. Are there more than these? Oh yeah, there's a ton more. But I choose these because those are the easiest to recognize. So recognize and then apply the proper names as well as the classification. Last category is prostaglandins. If I give you this picture right here, automatically you know you're dealing with a prostaglandin. This is a non-saponifiable lipid, and it has a really unique structure. Bottom line is, these are short-range messengers. So think of these as short-range hormones. How short-range? They just affect local areas. Some of them affect cell-to-cell. That's how short-range they are. This is an example of a hormone which affects constriction and dilation although I misspelled that. And this was actually used as a alternative to Viagra. The company didn't do so well because this is a short-range hormone, so they had to use this. In order for a male person to use this, they would have to use a needle into, yes, that area, or they'd have to use this as a penile suppository. Obviously, the company didn't do too well marketing this particular drug. The prostaglandin name goes by PGE1. If I give you the structure, recognize it as a prostaglandin, and that concludes our fatty acid lipids.